Hi everyone, so to be able to identify the different types of carbohydrates using this specific chemical test. So these are my group mates. I have Sales Aubrey, Saad Nicole, Sakayan Kailin, and me Santos Aldrin. In this report, what we are going to tackle, we will tackle about carbohydrates identification with a series of classic tests like Molish, Biles, Seliwanov, Benedict, and Barfoed, and of course the Tollens test.
Molish test is a chemical test that is used to detect the presence of carbohydrates. To perform the Molish test, you will need pipette, test tubes, test tube rock, test tube holder, Molish reagent, and concentrated sulfuric acid.
Unlike most other biochemical tests like Benedict's, Fehling's, Tollens, Barfoed's, Seliwanov's tests, etc., that are used to detect and distinguish specific types of carbohydrates such as reducing sugars from non-reducing sugars or ketose sugars from aldose sugars, the Molish's test is a biochemical test to detect the presence of carbohydrates in general. All carbohydrates, including monosaccharides, disaccharides, and polysaccharides, except trioses and tetroses, show positive reactions, although monosaccharides give a more rapid positive test as compared to disaccharides, oligosaccharides, and polysaccharides, which react slower because of the additional time taken for these complex sugars to break down into their constituent monosaccharide units through acid hydrolysis. Trioses and tetroses do not have the necessary 5 carbon atoms for furfural formation, so they do not give a positive test for this reaction. We'll come to the chemistry of Molish's test a bit later to understand this. Pentose sugar containing nucleic acids and also glycoproteins also give a positive reaction, as all these compounds are eventually hydrolyzed to monosaccharides by strong mineral acids. The Molish's test is in essence comparable to the rapid furfural test or the Seliwanov's test because they all involve acid dehydration of monosaccharides to form furfurals or their derivatives, and the eventual condensation of these furfurals with phenolic compounds. Molish's test is named after the Austrian botanist Hans Molisch, who invented the reagent and the test. Molish's reagent consists of a phenolic compound called alpha-naphthol dissolved in ethanol or other appropriate solvents. Make sure to watch my video on how to prepare Molish's reagent by clicking on the link given in the screen right now or the link given in the description below. The test is based on the underlying principle that when a concentrated acid is slowly added to a mixture containing Molish's reagent and a carbohydrate sample, a series of dehydration and condensation reactions occur between the acid and the carbohydrate sample on the one hand and the reagent and the carbohydrate sample on the other hand. The end result is the formation of a deep purple to violet colored complex, which is seen as a deep colored ring at the interface of the acid and the reagent carbohydrate mixture. If the monosaccharide in the sample is a pentose sugar, furfural is formed as dehydration product, which then undergoes a series of condensation reactions to finally yield a purple-colored complex. If the sample contains a hexose monosaccharide, then 5-hydroxymethyl-furfural is formed as dehydration product instead of furfural. The rest of the reactions proceed the same way as in pentose sugars to yield a purple ring. However, if disaccharides, oligosaccharides, or polysaccharides are present in the test sample, the concentrated sulfuric acid first hydrolyzes all the glycosidic linkages in these sugar molecules to yield monosaccharide units. The hydroxyl groups of these monosaccharides are then removed in the form of water molecules in a series of triple dehydration reactions to form aldehyde-containing furfurals in the case of pentose monosaccharides or 5-hydroxymethyl-furfurals in the case of hexose monosaccharides. These furfurals in turn react with 2 molecules of alpha-naphthol in a series of condensation reactions to give a purple to violet colored complex. One significant disadvantage of Molish's test, however, is that the test does not detect exclusively carbohydrates. Furfurals as such can yield positive results, Some organic acids can also give a positive result. In this video, we'll demonstrate positive malicious test using a few carbohydrate solutions as test samples. To perform the test, we'll need the following: malicious reagent. Click on the link given in the screen right now to know how to prepare the reagent. Concentrated sulfuric acid. Test sample consisting of 1% solutions each of the following: fructose as a representative of pentose monosaccharide, glucose as a representative of hexose monosaccharide, sucrose as representative for disaccharides, starch as representative sample for oligo and polysaccharides, and distilled water as control sample. 5 clean, dry, identical test tubes, test tube holder, test tube stand, 7 graduated droppers or pipettes of 1 or 2 ml capacities. Take the 5 clean, dry, appropriately labeled test tubes in the test tube stand. Using 5 different droppers or pipettes, add 2 ml each of the carbohydrate samples and the control sample to the 5 tubes. The tube containing distilled water will be the negative control for this experiment. Now, using a different dropper or pipette, add 2 to 3 drops each of malicious reagent to all the 5 tubes. Mix the tube contents by gently swirling. Using a different dropper or pipette, gently and slowly add 1 ml each of concentrated sulfuric acid along the sides of the tubes so that 2 distinct layers are formed in each tube. Now observe any color change at the junction of the 2 layers in all the tubes. Appearance of a purple ring at the junction of the 2 liquids indicates the presence of carbohydrates. As expected, this purple ring is formed in all 4 tubes containing the test samples because they are all carbohydrates with 5 or more than 5 carbon atoms in their constituent monosaccharide units. The negative control tube, as expected, shows a negative reaction as well. So this is all about the malicious test for carbohydrates.
For the explanation of the table, control resulted negative because there is no purple or violet ring formed at the junction. However, glucose resulted positive because there is a clear violet ring formed at the junction. Fructose also resulted positive because there is a distinct violet ring appeared at the junction similar to glucose. Sucrose resulted positive. There is a violet ring developed at the interface, though less intense. Starch also resulted positive. Starch produced a violet ring as well, but it is developed more slowly and was not sharp like the others.
Bial's test is used to identify pentose and distinguish them from hexoses. In this experiment, arabinose, which is pentose, produce a blue-green color when react with Bial's reagent. On the other hand, fructose and hexose only produce a yellow-brown solution. This result proves that Bial's test is specific from pentoses, since only they give a positive blue-green color. Bial's arsenal materials are arabinose, fructose, glucose, Bial's reagent, pipette, labeled test tubes, and measuring cylinder. The differences in visible color changes occur because sugars have different chemical structures and reducing properties. Arabinos react positively, producing the expected blue-green color, while glucose and fructose did not give the same reaction. Under these conditions, this shows that not all monosaccharides behave the same way in biochemical tests, and color changes are used as indicators to identify the presence or absence of certain functional groups.
The Seliwanov test distinguishes between aldose and ketose sugars. A positive result is indicated by a red color upon heating with Seliwanov reagent. Materials that we will be needing is test tube stand, test tube holder, beaker containing the sample which is the H2O water, measuring cylinder, and Seliwanov reagent.
We are going to perform the Seliwanov test. The apparatus used for this test are test tubes, test tube stand, test tube holder, measuring cylinder, and a beaker containing sample. The reagent used for this test is Seliwanov's reagent. Take a test tube and hold it with a test tube holder. Add 5 ml of Seliwanov reagent. Add 1 ml of sample. Mix the contents well and boil it for approximately 30 seconds. Shake the test tube while boiling it. Cool the test tube under tap water. There is a formation of a cherry red color.
Based on my observation, the result of the Seliwanov test is the color is red colored complex. Ketose sugar or fructose react immediately with mild acid to give furfural derivatives. These furfural derivatives combine with resorcinol to give cherry red colored complex. Ozazone test is for reducing sugars. Ozazone are compounds formed when the reducing sugar reacts with excess of phenylhydrazine. The materials that we will be needing on the ozazone test are the test solution of glucose 1%, phenylhydrazine hydrochloride 0.2 grams, sodium acetate 0.1 grams, and 5 drops of glacial acetic acid.
Reducing sugars. Sugars which are aldoses or containing hemiacetal on the anomeric carbon of the sugar is called reducing sugars. Ozones are organic compounds formed when reducing sugars react with excess of phenylhydrazine. Ozones are peculiar with each different reducing sugar. They have a characteristic crystalline structure which can be studied under the microscope. Materials required: the test solution, phenylhydrazine hydrochloride in excess, sodium acetate, and glacial acetic acid. Start by taking 5 ml of the sample solution. Here I have 1% glucose. You can use any reducing sugars like maltose or lactose. Into that, we will be adding 5 drops of glacial acetic acid. Care should be taken while handling glacial acetic acid as it is corrosive. Now we will be adding 0.2 grams of phenylhydrazine hydrochloride. Use gloves while handling phenylhydrazine. Now we add 0.1 grams of sodium acetate crystal. After adding all these compounds, we mix the contents well with the help of a glass rod. After mixing it well, we will keep it in a boiling water bath for 25 to 30 minutes. What is happening here is two separate reactions. Firstly, the glucose reacts with phenylhydrazine, giving glucose phenylhydrazone by elimination of a water molecule from the functional group. Next step, one equivalent of glucose phenylhydrazone reacts with two equivalent of phenylhydrazine. That's the reason for excess phenylhydrazine. The first phenylhydrazine oxidizes the alpha carbon to carbonyl group, and the second phenylhydrazine involves the removal of one more molecule of water with the new formed carbonyl group of that oxidized carbon, forming a carbon-nitrogen bond. The alpha carbon is attacked here because it is the most reactive carbon atom. This figure is the complete reaction in a nutshell. Okay, now we are back to the reaction. After 30 minutes, we take it off from the boiling water bath and cool it under running tap water. We then observe the crystals formed under a microscope in high power field. The following are the images which I captured using my microscope. The needle-shaped crystals indicate the reducing sugar is glucose or fructose. In the next figure, you see sunflower or petal-shaped crystals indicating the presence of maltose. And the last figure is lactose, forming hedgehog or powder puff-shaped crystals.
Benedict test is a chemical test used to detect the presence of reducing sugars in a solution. For Benedict's test, you'll need pipette, test tubes, test tube block, test tube holder, beaker, burner, and Benedict's reagent.
Hello everyone, today we are going to discuss Benedict's test. Benedict's test is a qualitative test done for the detection of reducing sugars in the given sample. So in today's video, first we will see theoretical aspect related to the Benedict test like principle, operational procedure, discussion of various viva question, and in the end we will see demonstration of Benedict's test. So please watch this video till the end. Benedict's test. All the reducing sugars will give positive Benedict's test. Reducing sugars are those sugars which have free aldehyde or keto group in their structure due to presence of anomeric carbon. Sucrose is an example of non-reducing sugar. So sucrose will give negative Benedict's test. Now we will see principle of Benedict's test. Reducing sugars under hot alkaline medium tautomerize and form enediols which are powerful reducing agents. Reducing sugars under hot alkaline medium tautomerize and form enediols which are powerful reducing agents, and these enediols will reduce cupric ions of Benedict's reagent to red cuprous oxide. So that is the principle of Benedict's test. Enediol is the enol form of sugar because two hydroxyl groups are attached to the double bonded carbon. This is the picture showing the principle of Benedict's test. As we have discussed, in the hot alkaline medium, reducing sugars are tautomerized to the enediol, and this enediol will reduce cupric ions of copper sulfate to the red cuprous oxide, and it will get oxidized to the sugar acid. So that is the principle of Benedict's test. The This is the picture showing the positive Benedict test if there is a presence of reducing sugars like glucose and fructose, and negative Benedict test if there is a presence of non-reducing sugar like sucrose. And if there is a presence of reducing sugar, then while applying heat to the test tube, you will observe sequence of color changes. So that sequence of color change is from blue to green to yellow to orange to brick red if there is a presence of reducing sugar. And if there is an absence of reducing sugar, then there will be no any color changes will be seen. So that is regarding the positive Benedict test and negative Benedict test. Now composition of Benedict's reagent. So Benedict reagent is consist of three components. First one is the copper sulfate, which provide cupric ions. Second one is the sodium carbonate, it provides alkaline medium. And the third one is the sodium citrate, it acts as a preservative. So that is regarding the composition of Benedict's reagent. Now we will see the procedure of Benedict's reagent, Benedict's test. In the procedure, in the test tube, take 5 ml of Benedict's reagent and add 5 to 8 drops of given sample or solution and apply heat to it for 1 to 2 minutes and observe the color changes. So that is regarding the procedure of Benedict's test. Now one frequent viva question which is asked in the exam, that why Benedict test is called as a semi-quantitative test. So while applying heat to the Benedict reagent, there is a sequence of color changes occur if there is a presence of reducing sugar from blue to green to yellow to orange and to brick red. So from the color of the solution, we can have idea regarding the approximate amount of sugar present in the solution or in the urine. If there is a presence of green color, that means up to 0.5% of reducing sugar is present in the urine. If there is a yellow color, that means 0.5 to 1% of reducing sugar is present in the urine. If there is a orange color, that means 1 to 2% of reducing sugar is present in the urine. If there is a presence of red color, that means greater than 2% of reducing sugar is present in the urine. So that is why this Benedict test is called as a semi-quantitative test, because from the color of the solution you can have idea regarding the approximate amount of sugar present in the solution. And this Benedict test is one of the common tests done for the detection of sugar present in the urine in the clinical laboratory. Another viva question which is frequently asked is when Benedict test is considered as a false positive. So substances other than reducing sugars like ascorbic acid, which is also called as a vitamin C, homogenesic acid, salicylic acid, formaldehyde, chloroform, lactic acid and nitrites will give false positive Benedict's test. So if there is a presence of any of this substance in the solution, then they will give false positive Benedict's test. Now we will see the demonstration of Benedict's test.
So we have already seen the principle of Benedict's test, which is done for the reducing sugar. Now we will see the procedure of Benedict's test. So for the Benedict test, you will be needing Benedict's reagent. So that is blue color reagent is Benedict's reagent. Then carbohydrate solution. Then test tube holder. Then round bottom test tube and the burner. Fine. So first you have to take around 5 ml of Benedict's reagent in the round bottom test tube. And then you have to hold the test tube with the help of test tube holder and then add around 5 to 8 drops of carbohydrate solution in it. After adding, mix it properly and then you have to heat it under the burner. So while heating it, you have to notice the color changes. If there is a presence of reducing sugar, then blue color will be changed to the green color. From the green color, there will be the formation of yellow color, and from the yellow, there will be the formation of orange color. And if you are heating it till the last, then this orange color will be changed to the brick red color. So this is, you can see already there is a formation of brick red precipitate is there. That means reducing sugar is present in the solution. And from the color you can also have idea about the approximate amount of sugar present in the solution. As there is a formation of brick red color, that means there is a presence of more than 2% of sugar in the solution. So this is about the Benedict test. Thank you.
Explanation of the table. Glucose resulted positive. The solution first turned green, green to yellow, yellow to orange, and finally formed a brick red precipitate. The explanation for this is glucose, a reducing sugar, a monosaccharide with a free aldehyde group, so it reacts strongly with Benedict's reagent. Xylose. Xylose resulted positive. A color change from green to orange, orange to red, was observed with precipitate. Explanation for this is xylose is also a reducing sugar, a pentose sugar to be exact, so it changes color just like glucose. Fructose. Fructose also resulted positive. The solution changed from green to yellow, yellow to orange, orange to red, ending with a reddish precipitate. The explanation for this is even though fructose is a ketose, in alkane solution, it rearranges to an aldose form, so it can also act as a reducing sugar. Lactose. Lactose resulted positive. The solution gradually changed to yellow to orange, producing a brick red precipitate. The explanation for this is lactose is a reducing disaccharides because one end is free to open up and react Benedict's reagent. Sucrose. The result for sucrose is negative. There is no change of color. The solution stays blue. The explanation for this is sucrose is a non-reducing sugar because both ends are locked in the glycosidic bond, so it can't react unless it's first broken down or hydrolyzed. From the experiment, it was observed that glucose, xylose, fructose, and lactose gave a positive result with Benedict's test as shown by the formation of orange to brick red precipitates. This confirms that they are reducing sugars. On the other hand, sucrose did not show any change in color and remained blue, which means it is a non-reducing sugar.
Barfoed's test distinguished monosaccharides from disaccharides based on their ability to reduce copper 11 ions in acidic solution. In this experiment, glucose rapidly produced a red-orange precipitate of copper 1 oxide within 3 minutes, indicating its strong reducing property as a monosaccharides. Maltose, however, react more slowly with a faint precipitate forming after heating, consistent with its classification as a reducing disaccharides. These results confirms that monosaccharides react quickly with Barfoed's reagent, whereas disaccharides show delayed reactions. Barfoed's test materials are maltose, Barfoed's reagent, glucose, test tubes, measuring cylinder, and pipette. Maltose, a reducing disaccharide, gave a weak or delayed red precipitate after 7 to 10 minutes, showing it reacts slowly in Barfoed's test. Glucose, a monosaccharide, produced a red precipitate within 2 to 5 minutes. Confirms that monosaccharides reacts faster than disaccharides in reduction tests.
Tollens test, also known as the silver mirror test, is a chemical test used to distinguish between aldehydes and ketones. It relies on the oxidation-reduction reaction between an aldehyde and Tollens reagent, where the aldehyde is oxidized to a carboxylic acid and silver ions in the reagent are reduced to metallic silver, forming a silver mirror on the reaction vessel. To perform the Tollens test, you'll need silver nitrate solution, dilute sodium hydroxide solution, ammonium hydroxide solution, benzaldehyde, clean and dried test tubes, hot water bath, and droppers.
This video demonstrates how to use Tollens reagent to test for aldehydes. This video first demonstrates the preparation of Tollens reagent. Two tests will then be carried out. One shows a positive result, and the other shows a negative result. Preparation of Tollens reagent. Add 2 ml of 0.3 molar silver nitrate solution into a clean test tube. And then add 1 to 2 drops of 0.3 molar sodium hydroxide solution. Brownish precipitate of silver oxide will be formed. Add a 3 molar aqueous ammonia solution drop by drop with a dropper, until all the silver oxide precipitate is just dissolved. Do not add excess amount of the ammonia solution. The solution obtained is aqueous diamine silver solution, which is also known as Tollens reagent. Add a small amount of sample to the Tollens reagent. Shake the mixture gently, and then stand it on the bench for 10 minutes. Observe if there is a silver coating formed on the inner surface of the test tube. If there is no observable change, warm the mixture in a warm water bath for 5 minutes. If the sample is an aldehyde, a positive test result will be obtained. If the sample is not an aldehyde, the test will give a negative result. It is important that Tollens reagent should be freshly prepared. Prior to disposal, the reagent must be acidified immediately after use. The addition of acid prevents the formation of explosive silver nitride.
For the explanation of the table, glucose resulted as its aldehyde group undergoes oxidation to produce a carboxylic acid, which makes the test positive. For the explanation for this is because the silver ions are reduced to metallic silver, which creates a mirror on the reaction vessel or the test tube, while the aldehyde group in glucose is oxidized to a carboxylic ion by the Tollens reagent. The cyclose didn't give a positive reaction. The reason for this is that the reaction of the silver ions in the reagent are reduced to a metallic silver, which subsequently forms a mirror on the test tube, while the cyclose includes a free aldehyde group that can be oxidized. The result of sucrose does not react to produce a silver mirror, signifying a failure. The reason for this is because the sucrose, a disaccharide, inhibits the creation of a free aldehyde group required for the reaction with Tollens reagent, because it contains both anomeric carbons engaged with a glycosidic bond.